Okay, I am very delighted to introduce Dr. Billy Mills. And Billy did his undergraduate work at UVA, getting a degree in biology and biochemistry, and then did his PhD right here at Johns Hopkins in biological chemistry. And he is now an assistant professor at Mount St. Mary's. So we're going to talk all about the fascinating world of gene silencing and microRNAs, and I think a very under talked about area in the mitochondria in particular. So thanks so much for being here. We're excited for your talk.
Awesome. Thanks so much for the invite. I'm very happy to be here and share with you a little bit about mostly the work I did in graduate school and the things I'm continuing on now. As was said, my name is Billy. I'm currently teaching and have the privilege of teaching at Mount St. Mary's in Emmitsburg, Maryland. Let's see here. All right, so what I'll be doing today is I'll be telling you a little bit about what microRNAs are in the first place, how they were discovered since they were recently awarded the Nobel Prize, then specifically how we study them in the lab, the techniques that I use to study them, then what I learned in graduate school about what these little molecules are doing inside of mitochondria, and then I'll end with a little bit of perspective on what I think would be great next steps to follow up on with these findings. So first, what are they? To begin, I always like to start with this idea that you may have heard of before called the central dogma of molecular biology that says that the DNA in the nucleus of all of our cells is transcribed to make messenger RNA or temporary copies, mRNA, and that messenger RNA is translated to make proteins. And this is the general flow of information in our cells. And what's unique is that that same DNA in each one of our cells is able to give rise to all of the different cell types we see in our body. Skin cells, muscle cells, immune cells, neurons, all cells. And as you can see just from these cartoon pictures, each of these cells has really unique morphologies, shapes, functions, processes that go on inside of them and outside of them. And then the question is, how does our DNA, which is the same in every cell, give rise to all of these unique cell types? And the way it does that is that this central dogma is not so straightforward. There are lots of things that are acting on this pathway at each level, from DNA down to protein. As the color of the arrow shows, some of these activities enhance whether or not this process happens, and others turn it down. And the one in particular, of course, that I'll be talking about today are microRNAs, and they work specifically at the level of the messenger RNA, where they are usually preventing messenger RNAs from being translated into proteins. And so I'll give a little bit of background now for the history of their discovery before we move into some of the techniques that I use to study them. So like I said, this question of how is it that one genome is able to give rise to so many different cell types in our body, from neurons, muscle cells, epithelial cells, and immune cells, all from the same starting material. And this question was originally picked up on by lots of scientists, but the two in particular that I'll be talking about are Victor Ambros and Gary Ruvkun. And they were doing their studies in a model system called C. elegans, a really small microscopic worm. And this is a great model system to use. For one, it has all of these different cell types. It has neurons and gut cells and muscle cells, all arising from the same starting genome. So it's a great model system to work with. It's cheap to work with in the lab and study. And one of the things that first piqued their interest was that there were some mutants of this worm that had interesting morphologies. And you can see here the Lin4 mutant is the one that I'll be spending the most of the time talking about. Specifically, this is the exact name that they use. But it might be hard to see, but between the normal and the mutant form, they might be roughly the same size, but you might be able to appreciate these little speckles in the inside of the worm that are a result of a mutation in this Lin4 gene. Another mutant, the Lin14 gene, very obviously has a defect much smaller than the normal form. And the question these scientists had was, what is giving rise to these different phenotypes based on their mutations? These discoveries were happening simultaneously. These two scientists did a postdoc together, and then they went on to their own separate labs where they were studying these pathways. Victor Ambros was studying this Lin4 mutation, and what was originally hypothesized, as was known in the field at that time, was that most mutations like these were probably arising from mutations in messenger RNAs, which are leading to defects in proteins that are then causing these phenotypes. So they were setting out to see, what is this Lin4 gene? What protein does it encode? And when they did their genetic screening and they tested this, what they found was kind of striking, that the gene that encoded whatever this Lin4 thing was short, was really short. Most messenger RNAs are thousands of bases long, but when they rescued this mutant, they added back the gene, and what they saw was that there were two pieces that were expressed when they reintroduced this. They called them Lin4L for Lin4 long, and Lin4S for Lin4 short. You can see their sizes based on this scale, roughly 20 or so bases and roughly 50 or so bases, but not the thousands of bases you would expect for a messenger RNA. So that was pretty striking. Something small is responsible for this phenotype that they observe here. Simultaneously, the other scientist, Gary Ruvkin, he was studying this mutant as well, the Lin4E912 mutant, and what they found was that when they studied this mutation and they tracked the levels of what was known to be a protein, Lin14, they found that in the wild type, when you go from early to late development, the levels of this protein decreased drastically. But in the worms that had a mutation in Lin4, which is a completely different gene from Lin14, when they go from early to late and have this mutation, the levels of this protein don't go down, which would suggest that there's some interaction or relationship between Lin4 and Lin14, which is interesting because these two are not encoded together anywhere close to each other in the genome, but they seem to have some interaction. And specifically, it looks like Lin4 is turning down Lin14, and then when you lose Lin4, you get lots more Lin14, as is shown here. When they looked at the sequence of these RNAs, specifically these short RNAs, what they found was rather interesting. There was complementarity between the Lin4 small sequence and the Lin14 messenger RNA that encodes for this protein. As a reminder, DNA and RNA can form base pairs with one another. In RNA, C always pairs with G, and U always pairs with A. And so when they saw this, it was interesting, and it suggested to them that maybe the fact that Lin4 can form these base pairing interactions with Lin14, maybe that's how it regulates the messenger RNA. Maybe by binding to it, it prevents it from being translated. And the mechanism would take many more years to work out, but at least this was the preliminary discovery that some short RNA could regulate a different protein's expression. A follow-up to this, also in Gary Ruvkin's lab, was to look to see, this is an interesting phenomenon, but is it only true of worms, or is this something that's conserved across species? And they went and they found that there was this short RNA Lin4, but they also found another small RNA called Let7. Then they looked to see, do we find these two genes or conserved sequences of them in other species? They looked to humans. They couldn't find the Lin4 sequence, but they did find the Let7 sequence. So there was a conserved short RNA sequence between worms and humans. And then when they looked broadly across species, they found that, and now we know better, that almost all eukaryotes express this Let7 microRNA. And just a cool story, this scientist, Amy Pasquinelli, who's the first author on the paper that made this observation, she actually came and spoke at Hopkins while I was in grad school, so got to meet her about 5 years ago. And so this is now the model that they had when they made these discoveries, that in our genomes, there are the genes that encode for messenger RNAs that we normally think are getting translated to proteins. But our genomes also possess genes for small or short RNAs, later called microRNAs, that bind to those messenger RNAs and prevent them from being translated into proteins. And that this interaction is what gives rise to some of the interesting things we see in biology, like how organisms develop, different cells arising from the same starting genomes, and even how we can convert between diseased states like cancer and healthy states. Just last year, these two scientists were awarded the Nobel Prize for, quote, their discovery of microRNA and its role in post-transcriptional gene regulation. Post-transcriptional in that microRNAs act just after DNA has been transcribed into messenger RNA. All right, so now I'll fast forward, now 30 years since their discovery in 1993, we know a lot more about how they're made, what they look like, and what they do, so I'll give a little bit of background on what we know now. This is a busy slide, but just to highlight that they have a very unique process of being synthesized. Most messenger RNAs get transcribed and then spliced and leave the nucleus. MicroRNAs undergo something similar to produce a different molecule. So they're transcribed in the nucleus, they make a primary or a pri-microRNA. It's processed by a complex called the microprocessor. It's cleaved at the base of this stem to form the pre-microRNA that then leaves the nucleus. And once it's in the cytoplasm, this pre-microRNA can be processed by even more enzymes and proteins. And ultimately, through this cleavage, one strand of this stem gets loaded onto a protein called argonate. And argonate is the key protein component of what will become the RNA-induced silencing complex, or RISC. And the sequence of the microRNA in the RISC is what targets this complex to its target messenger RNAs, right? If there's compatibility or complementarity between the sequence of this microRNA and this target, it will bind and theoretically prevent that messenger RNA from being translated. If we zoom in here at a cartoonish view of what this complex looks like, you can see a single microRNA bound to and theoretically inhibiting the expression of a target messenger RNA. And the ability of a microRNA to bind to a messenger RNA is determined by this complementarity. And specifically, there are two regions that are really important. The first, and probably the most important, is what's called the seed region. About 6 or 7 bases near one end of the microRNA that are thought to be the main determinant of specificity. That is, if these match between the microRNA and the target, it's most likely a target of the microRNA. But there are other regions towards the other end of the microRNA that can enhance or aid in this binding, but the main determinant of specificity is thought to be this seed region. With that in mind, when scientists understood this, they asked the next question, well, what is it then? How can we then determine what the targets of microRNAs are? Their thought was, if the seed region is doing most of the specificity, all we have to do is look for seed sequences or matches in target messenger RNAs. And so they would just scan and look to see where does this seed sequence bind in all the messenger RNAs in our cells. And that's a decent first way to do this, but there are some limitations. Biology is complicated. And again, busy slide, but just showing you some examples of why just predicting interactions could be insufficient. So here's a lettered view of what that interaction looks like, a really strong seed sequence, some 3' sequence that's helping. But what's now known is that there are sequences or interactions between microRNAs and messenger RNAs that have really weird looking seed sequences. They have bulges and mismatches in that important window, but they're still real interactions. So if all you did was look for seed matches, you would miss unique interactions like this. It was originally thought that the main site where microRNAs were binding was in this region of a messenger RNA called a 3' untranslated region, or 3' UTR. And so those original prediction tools just simply looked for this interaction, microRNAs and 3' UTRs. What we now know is that microRNAs can actually bind anywhere in a messenger RNA, from the 5' UTRs, introns, and even exons and even introns. And so if you just looked here, you would miss those other interactions. When these methods do this prediction, they start with what's called sequencing. They just simply determine the sequence of all of the RNAs in their sample. And when they do that, you have to break open the cells, lyse the cells open, collect all the RNA, sequence it, and then determine the letters of A's, U's, C's, and G's in their sequences. When you do that, you lose the ability to see what was happening inside the cell, what was interacting with RUD. Because when you break open the cell, everything gets kind of jumbled up. And so you might be predicting interactions in your sequencing data that weren't actually happening in your biological system. For example, RNAs are unique molecules in that they can fold and interact with themselves, called RNA secondary structure. You can imagine there might be a potential site here, shown in the light blue, but it's folded and not accessible. If you sequenced, you would be breaking this open, unfolding everything, and you would be predicting an interaction, but biologically that site wasn't available. There are also proteins called RNA binding proteins that do just that. They bind to RNAs. They can block sites from being accessible. But again, in sequencing data, you wouldn't know that that was happening inside the cell. That argonate protein, that's the key protein component of the RNA-induced silencing complex. It's not just a static protein. It can undergo post-translational modifications. There are other proteins and factors that can bind to it that change how it behaves. And that, again, is lost in sequencing alone. And finally, this is really relevant for my graduate work, we were studying the nervous system. Neurons are these really large, highly compartmentalized cells. You can imagine you might have a microRNA in one part of the cell and the messenger RNA in the other part of the cell, and biologically they were not interacting. They couldn't. But in your sequencing data, when everything gets split open and jumbled up, you wouldn't know that. You might predict an interaction for two things that weren't even in the same part of the cell. So for all of these reasons, scientists needed a better method than just predicting interactions. And here enters the technique that I use. This was developed by several labs about 10 plus years ago. And the key step in this protocol is that when you have this complex, you want to preserve this interaction as much as possible. And so what they do is they use an enzyme to physically stick together or ligate the microRNA to its target within this complex. And now, once they're stuck together, you don't need to predict interactions, right? They're stuck together, so you know exactly what was interacting with what inside of the cell. And it's this method and a modification of it that I developed in graduate school that I use to study microRNAs. All right, so now that we have the background for what they are, how we study them, now I'll tell you specifically about the protocol that we use in the lab to study them and how we generate those molecules. I'll be using this cartoonish view again, reminder, a single microRNA interacting with a single messenger RNA in the context of this protein called argonaute, where the key processing or the key interaction step is occurring. And I'll walk you through each of the steps that we do. You'll see the names of the enzymes appear in the bottom right. If you're curious, they're there for you, but just the main thing is to see how these two RNAs are processed. So the first step in the protocol is to use ultraviolet light to stick everything together. Scientists have found that if you expose protein and RNA to ultraviolet light, if they're close together, they'll actually form bonds that hold them together permanently. And so when we expose our samples to ultraviolet light, I denote with the X, now we formed a bond that will hold these RNAs to the protein for the rest of the procedure. The next step that's really important is that we ultimately want to stick this end of the messenger RNA to this end of the microRNA. Messenger RNAs, as I mentioned before, can be thousands of bases long. And so this end might be really far away, much farther than is shown in this cartoon from the end of the microRNA. And so if we want this to work, we have to cut back this RNA so that these two ends are close together. And that's what that enzyme does. If we want to ligate them together, we're going to have to isolate these complexes from the rest of the cellular material, right? I'm just looking at one protein among hundreds of thousands in the cell. And so if I want to study these RNAs, I'd like to get rid of all that other stuff and just look at these. So I'm going to isolate these complexes using an antibody that binds to argonaute and a magnetic bead so I can suction these to the side of a magnet and wash away everything that I'm not interested in. Then I just have to do some chemistry on the ends of these molecules so that they're compatible. And now here's that critical step where I use an enzyme called a ligase to stick these two strands together. And here we formed what we call a chimeric RNA. It's a single RNA strand now, but it has two things within it. It has a microRNA and a messenger RNA in the same strand. And this is really beneficial because I don't need to predict what was interacting. I can just see literally what was interacting. I'm going to do some more chemistry to prepare these ends to ultimately add what's called an adapter or a linker. If you ever do sequencing experiments, these are instruments in which you basically add your molecules to a machine and those molecules get stuck onto the machine. So you can kind of think of this as a hook that the machine grabs onto to hold these sequences in place so that they can be sequenced. And so we're going to be adding links to both ends and preparing them chemically for this to happen. These next few steps, the ultimate goal of them is just to get the RNA separate from the rest of the protein. And so we'll run these on a gel, cut them out by their proper size, and then we're going to basically digest away all of the protein so we're just left with the RNA. And it looks like this. Again, microRNA stuck to a messenger RNA with a little adapter or a hook on the end. I have to add a hook or an adapter to the other side for this sequencing reaction to work. When we sequence, we can't sequence RNA, we can only sequence DNA, so I use a process called reverse transcription to turn this RNA back into DNA. It's called cDNA because it's complementary to the RNA that was used to make it. And then when I want to do sequencing, I don't need just one molecule of these. I need several of them or a few of them so that the machine can detect them. And so we use PCR to amplify this region and get lots of copies of them. And now these are the molecules that we send away to a sequencing facility that sequences them for us and then gives us back those sequencing data. And just to show, if you were curious what this looks like in the lab when we're optimizing all of these steps, this took many years to get to work well in the lab, but this is kind of what those raw data look like that proved to us that this technique worked the way that we thought it should. The first step I told you was to use ultraviolet light to form cross-links between the RNA and the protein. And when we do that, what's interesting is that argonaut normally has a weight of about 100 kilodaltons, and you can see its intense band here. And when you increase the amount of UV that you expose to, it almost looks like it's disappearing, like it's being degraded or something. But what's actually happening is that this argonaut, by itself, normally 100 kilodaltons, is now getting RNA stuck to it that brings along more mass and size. And so it's not disappearing, it's just getting shifted to a higher and higher weight. And we can prove that because if we stick radioactive isotopes of phosphorus to these RNAs and visualize them with autoradiography, here they are. The black signal here represents the radioactive isotopes. And you can see that there are none when you don't cross-link, but when you cross-link, you then just shift these complexes to a higher weight. One of the key steps was to chew back the ends of the RNA so that these two are close together. That using an RNase enzyme, we have to make sure we get the right amount in there. You can imagine if you don't chew back enough, these ends won't be close enough together. And if you chew back too much, you could actually just get rid of everything. So you have to find that sweet spot of how much enzyme to use. And that's just the row or the lane here that has the most signal. And so we got the correct concentration there. And this final figure, if you put all those pieces together, use the right amount of ultraviolet light and enzymes, you can see that as we just increase the amount of starting material, where this signal starts to arise is where these complexes are. And so this is the region of the gel that I cut out, isolate those RNAs, and then prepare them to be sequenced. And on the end here are two negative controls, right? If you don't add this enzyme to chew them back, you get nothing. And if you don't add the enzyme to ligate them together, you also get nothing. So this kind of shows us this is the optimal conditions to do this experiment in. All right, so with this method, we started getting back our sequencing data, and we had been reading methods in the past that had done this experiment, and we tried our best to replicate exactly what they were doing, but had a lot of trouble. So we worked really hard to improve the method to get it to work in our lab. And we spent so much time, we wanted to see, did we actually improve anything at all? Are we just doing the same thing they're doing, or is this method any better than the ones that came before it? And so we compared the percentage of our sequencing data that actually was interesting and unique. That's a measure for how well the experiment worked, is how much unique data there is in your data set. And this method here, this column at the end, is the method I developed called Chimera-seq. You can see that it's at least twice as good as the methods that came before it, at least in terms of what percentage of the sequencing data is unique and interesting. And so we were reassured that, yeah, it was worth all of the trouble. We could get a lot more interesting data back. All right, just a comment here on what happens now that we have the data back. What happens computationally? How do we analyze this data? So this is kind of the second hurdle we had to cross after the biochemistry was working. When you get the data back from the sequencing lab, there's more in that data than just the thing you're interested in. So I'm really interested in just this microRNA target RNA interaction. But in order to get it sequenced, I had to add all this extra stuff to it to get it to work. So if I want to study this stuff in the middle, I have to computationally remove all of this extra stuff, and then I have to individually study these two regions. And so another part of my graduate work was developing a computational tool to do this. So if you got this type of data, you could run this through our tool. It's called Scrap. And what it does is it removes all this extraneous information. It isolates just the region that you're interested in studying. It takes the microRNA part and aligns it to a database of known microRNAs to find out which one is there. And then it takes the rest of your data and it aligns it to the genome of whatever species you're working with. We often work with human cells or mouse cells, so we would align to their genomes. And then it brings this information together and it says, look, this microRNA was interacting with this gene from the genome in your species. And so this is the tool that we use to analyze the data once we get it back from the sequencing facility. Okay, so now that we have our techniques down, our biochemical wet lab techniques and our dry bench computational techniques, what did we actually find when we look through this data? And this is where the story of mitochondria comes into play. So when we were looking through our data, what we found was rather striking. First, we wanted to see, is there any difference in where or how microRNAs are binding across the genome of the species? So this was done in mouse. We were studying mouse brain for this experiment. And this was just a broad analysis. Do microRNAs bind to genes that come from these chromosomes with any higher frequency than the others? And this data suggests, no, it seems to be binding to genes pretty robustly across the genome. All of this is normalized to the length of the chromosome, so you're not biasing this for bigger chromosomes. There seemed to be no preference, with one exception. When we looked at the data and had somebody else look at the data, they said, did you notice there's a lot of mitochondrial chromosome reads in here? And when we put those in this analysis, we were rather taken aback that there are a lot of interactions with the mitochondria, with genes that are encoded on the mitochondrial genome. Many orders of magnitude higher than what we were seeing for the other chromosomes. So this was the first piece of evidence that kind of piqued our interest that maybe there's something interesting going on here. Not something that had really been reported in the literature, that microRNAs could interact with mitochondrial transcripts inside of the mitochondria. As a reminder, each one of the cells of your body has DNA in two places. Most of it, 99.9% of it, is found in the nucleus of your cell. These are those chromosome images you're probably used to seeing. But you do have a little bit of DNA that you inherit from your mother because you get your mitochondria from them. And these circular DNAs actually encode for a few dozen genes that are important for mitochondrial function. Mitochondria do use genes from our nuclear genome to function properly, but they also encode their own genes. So when you hear me refer to nuclear encoded genes, I'm referring to genes that are found here in the nuclear genome that get transcribed and then work in the cytoplasm. And when I refer to mitochondrially encoded genes, I'm talking about genes that are encoded on this little circular chromosome inside the mitochondria that get transcribed here. So mitochondrially encoded would be found in the mitochondria, and most of the nuclear encodeds are found in the cytoplasm. So the first question we asked was, okay, if they seem to be interacting with the mitochondrial genes, are there any differences in which microRNAs are doing this? Are the microRNAs that interact with nuclear genes the same or different than the microRNAs that interact with mitochondrial genes? So our first analysis was just to look at the distribution of microRNAs. So here we took all of the reads that mapped to the nuclear chromosomes, and we asked which microRNAs are binding, and we then put them in this pie chart. Just to comment here, I mentioned that the seed sequence is the most important part of the microRNA for determining the specificity. There are actually different microRNAs that can share a seed sequence, and we refer to those as a family. So if different microRNAs have the same seed sequence, they're part of a family. Let7 is a family of microRNAs that share a seed sequence. miR-124, for example, is its own microRNA. It's not part of a family. It's an individual microRNA. So that's why I say small non-coding RNA or microRNA/family here. So what's interesting is miR-124 is a neuron-specific microRNA. It's most enriched in neurons, which was reassuring to us because this study was done in the mouse brain. So good that we see it very abundantly here. The next most abundant was the Let7 family, which was interesting for our lab because this was a microRNA that we had studied. And then you can see the rest fall out here with the other category. So the question we had was, if this is a unique biological phenomenon, it would be cool if there was a different distribution of the microRNAs that interacted with the nuclear transcripts and the microRNAs that interacted with the mitochondrial transcripts. And we were kind of surprised to see that there seems to be a really big difference. We thought maybe they'd be similar, maybe microRNAs just bind to whatever's available, but it seems that there are some microRNAs that actually interact more with mitochondrial transcripts than they do with nuclear transcripts. Here you can see this Let7 family, which made up maybe 7-8% here, makes up almost 50% of all of the interactions that we see when we're looking at mitochondrial transcripts. And so this was our first indication that this was something really interesting that we should look into more deeply. So the next analysis we do is called a volcano plot. And just to orient you of how this plot is set up, the color and position of these circles indicates how statistically significant and to what extent they are enriched. These circles in the top right quadrant here, these are microRNAs that seem to bind to mitochondrial transcripts more than they do to nuclear transcripts. And dots in the bottom left are ones that bind to nuclear transcripts more than they bind to mitochondrial transcripts. The further left you go and the further right you go, the more significant that enrichment is, how many fold change higher. And then the more vertically you go on the y-axis is how statistically significant that enrichment is. So things that are in the really upper right would be things that are really highly enriched for mitochondria, and things in the top left would be things that are really highly enriched for the nuclear genes. And then things in the middle or the bottom are not statistically significant. So these here, of course, the dots are unlabeled, so we had to go in and look at what they were. One of them that was, I think, very interesting that I want to follow up on further was this microRNA called miR210-3P. It seems to be the microRNA that's the most statistically significantly enriched for binding to the mitochondrial transcripts. And I knew nothing about this microRNA, so when I looked into it further, this microRNA is one that's known to be induced by hypoxia. So when your cells are starved of oxygen, this microRNA's levels increase. And it's well known that this microRNA regulates mitochondrial function. And we're seeing that it's enriched in binding to mitochondrial transcripts. What's really cool is that, to our knowledge, no one had ever reported that miR210-3P regulates mitochondrial function by going into mitochondria and interacting with those transcripts. What all of these studies have found is that miR210 has targets from the nuclear chromosomes that impact how mitochondria function. But to our knowledge, like I said, I have yet to see a paper that says that miR210 regulates mitochondria by going into mitochondria and binding to those genes there. So that's something I would like to follow up more in my research, but that wasn't the microRNA that we were really following. So just a side note there. But at least reassures us that maybe that these data are significant if the microRNA that's most associated with regulating mitochondrial function is the most upregulated in binding to mitochondrial transcripts. But when we looked then at the 20 most abundant microRNAs, what we saw was very cool. All of the members of the LET7 family are enriched for binding to mitochondrial transcripts over nuclear transcripts. In particular, of all of the top 20 microRNAs, the only ones that are enriched for binding are the LET7 family. miR98 has a different name, but it's actually a member of the LET7 family. So you can see all of the LET7 family members are in this top right quadrant. So that tells us not only do LET7s bind with a greater proportion to mitochondrial transcripts compared to nuclear transcripts, they also seem to do so in a statistically significant way, as if they have a preference for binding there. And I have yet to talk too much in detail about this. This was the second microRNA ever discovered that I told you about, the one that was conserved across species. What's interesting about this microRNA is that it exists in a negative feedback loop with a protein called Lin28. Lin28 protein binds to the LET7 precursor and prevents it from becoming a mature microRNA, and the mature LET7 microRNA binds to the Lin28 messenger RNA and prevents it from becoming a protein. So these two are kind of working against each other during development and lots of other cellular processes. In early development, Lin28 is high and LET7 is low, and then later in development, LET7 is high and Lin28 is low. So this was interesting. This was actually the microRNA family that the lab I was in for grad school was really interested in studying. So it was really cool to see that this was implicated here. And you can see from the titles of these papers that several people have reported that this axis between LET7 and Lin28 is involved in metabolism, but again, no one had yet reported that it does so by binding inside of mitochondria to mitochondrial transcripts. So we wanted to look at it a little bit further. So I've showed you that they seem to have a preference for binding to mitochondrial transcripts, but the next question, of course, is which transcripts, which genes in the mitochondrial genome are the ones that the LET7 family or all microRNAs are binding. And so here, this track shows you from left to right all of the mitochondrial genes. There are about 24 of them. So if you take that circular chromosome and you split it up and you make it linear, we've just arranged all of the genes in a line like this. And you can see these peaks, the height of these peaks represents more microRNA binding. The taller the peak, the more microRNAs that are binding at that site. And if you look across the entire genome for the data that I generated from mouse forebrain, you can see there's a very obvious preference. The site that seemed most bound is right here, and you can zoom into that site here. What caught us off guard, though, is that this gene that's underneath of that site is not a messenger RNA. Normally, like I said at the beginning, microRNAs were thought to bind to messenger RNAs. This is actually a ribosomal RNA. You might remember ribosomes are those protein factories that synthesize proteins. They themselves are made up of proteins and ribosomal RNAs, or rRNAs. This gene, mtRNR1, MT just stands for mitochondria, but RNR1 encodes for the 16S ribosomal RNA. Sorry, the 12S ribosomal RNA. And so that's one of the components of the ribosome. So it's not unheard of. There have been reports that microRNAs can interact with ribosomal RNAs, but it's striking to us that of all the genes that could be bound in the mitochondrial genome, the ribosomal RNA seems the one that's most bound. This was so striking to us, we wanted to see if this is real, other researchers should have found this in their data. No one had reported it, but maybe they just weren't looking at this region of their data. So we went online. The NCBI Gene Expression Omnibus, the GEO, has data from experiments that have already been published. So we just looked for people who had done techniques similar to what we were doing and then ran their data through our scrap pipeline. Another group that did a very similar method to ours, this is the method that inspired me to try to improve it to Chimera-seq. When we ran their data through our pipeline, you can see a nearly identical result. The strongest peak is here on this gene RNR1, and then less binding throughout the genome. Their experiments were also in mouse forebrain, so I guess it makes sense that they look really similar. Yet another study did this technique not in mouse brain cells or neurons, but in mouse skin cells called keratinocytes. And again, less pronounced, but the most extreme peak in all this data is also on the RNR1 gene. And yet one more study, this one was now done in cardiac myocytes, so heart muscle cells from mouse. You can see again the most prominent peak is right over this gene RNR1. And so this was really striking to us that all of these data sets from mouse, using slightly different techniques, all observe microRNA binding at this particular gene, mtRNR1, or the 12S ribosomal RNA. And so what was awesome to us is when we were looking for all these papers that had published data that we could use for this, we wanted to then see, surely someone has mentioned this. This is so striking to us. Surely someone has made comment of this phenomenon. And one of the papers did. And I'll just read the quote. It says, interestingly, AGO2, which is argonaut, seems to prevalently interact with the 12S mitochondria-specific ribosomal RNA, RNR1. In myoblasts, which is one of the cell types they were using, which was further induced after differentiation to myotubes. So just a little bit about the biology of their system. They were studying how a premature muscle cell differentiates into a mature muscle cell. And what they observed was when they looked at the prematures and the matures, binding at this site increased when they looked at the matures versus the prematures. So not only does argonaut bind to this site, supposedly directed by microRNAs, it does so more as the cell develops. So as you become more mature as a cell, more and more binding occurs at this site. At least that's the conclusion that they are making here. But again, this was not the focus of this study. This study was studying a different microRNA called miR1, doing a different thing in the mitochondria. So they make comment here, but they then pursue miR1 a little bit further. Interestingly, in their study, they used a different method than I used, still studying argonaut, but they didn't do that chimeric ligation step. They did not ligate the microRNA to its target. So they actually don't know what caused argonaut to bind at this site. All they know that it does. That's why they just say argonaut seems to prevalently interact. So unfortunately, they don't, and we can't know from their data which microRNA was directing argonaut to this site. But those other methods all use the chimeric method, so we do know which microRNA was targeting it to that site. And when we looked in my data at this site, I'm looking at all of the interactions between a microRNA and this site in RNR1, and then asking which microRNAs are responsible for targeting that site. And as you can see, all of the LET7 family members here are what's targeting it to that site. So as a family, well over 50%, approaching 75% of all of the interactions between argonaut and this gene are directed by the LET7 family of microRNAs, which was really interesting to us. And I don't have the picture here, unfortunately, but if you look to see how they base pair, like I showed on the first slide of Lin4 interacting with Lin14, the interaction between LET7 and this site is like a beautifully standard microRNA target interaction. It has a really strong seed sequence and some complementarity sequence in the 3' region, so it looks just like a microRNA interacting with a target should. So all of that seems to suggest that it might be a true target. All right, so then once we have this information and these cool findings, where do we go next? What am I interested in studying from here? All right, so I'd like to just kind of tie together some data and some interesting findings from across literature and kind of try to tell a story about what might be happening here. So first, I had mentioned that Lin28, the protein, and LET7, the microRNA, are in a negative feedback loop. When Lin28 is high, shown here with this blue arrow, LET7 is low. And as cells develop, Lin28 levels go down, and therefore LET7 levels are allowed to increase. And so later in development, LET7 is high and Lin28 is low. This is a super well-studied system. Anywhere you look, this is the case for Lin28 and LET7 under normal conditions. Another trend that follows during development is that premature cells, before they become their mature forms, are highly proliferative. That is, they divide like crazy, but they're not very differentiated. So if a cell that's going to become a muscle cell is in its premature state, it divides and divides and divides, but it doesn't look like a muscle cell yet. As that cell matures and becomes more like a muscle cell, it divides less and less, but it starts to look more and more like a muscle cell. So we say it's more differentiated, but less proliferative, whereas in early development, it's more proliferative and less differentiated. And yet one more trend that follows during development is that early on in development, a cell in an organism relies mostly on glycolysis. And as a reminder from your metabolism lectures in biology, glycolysis is the metabolism that happens outside of the mitochondria. Oxidative phosphorylation is the metabolism that happens inside of mitochondria. So another way of potentially viewing this trend is that early on in development, cells rely more on non-mitochondrial metabolism, but as they mature and develop, they start to rely more on mitochondrial metabolism. And so all of these trends seem to hold together, and there's data that connects each of these rows of information too. As LET7 levels increase, cells become more differentiated and less proliferative, and they tend to rely more on oxidative phosphorylation, which is mitochondrial metabolism. Early in development, LET7s are lower, cells are less differentiated, and they rely more on glycolysis. So what I've been describing so far is development, normal, healthy development for cells and organisms, going from left to right. What's fascinating is that cancer cells seem to go backwards on this path. One of the hallmarks of cancer is that the levels of the LET7 family of microRNAs decrease, and usually decrease substantially. So you move backwards on this chain, you get less and less LET7. Cancer cells also revert a bit. They become less differentiated, but they divide like crazy, right? That's what makes a tumor. So they become more proliferative and less differentiated. And what's super fascinating is that they rely less on oxidative phosphorylation and more on glycolysis. So they're also working backwards in this chain. That phenomenon is so well studied, it has its own name. It's called the Warburg effect, that cancer cells, when they turn to be cancerous, they shift their metabolism from oxidative phosphorylation to glycolysis. So now the question I'm trying to ask is there a connection between these two? Is the fact that LET7 levels increase during development at the same time that cells start to rely more on their mitochondria, is it because LET7s might be going into mitochondria and accelerating their function or turning them on essentially? And this is supported at least by the fact that cancer cells do the opposite. When LET7s go down in cancer, they seem to be turning off their mitochondria and relying more on glycolysis. So those two opposite reactions both kind of support the premise that maybe LET7s go into mitochondria and they enhance mitochondrial function. But if you've been following along so far, that might give you pause, because when I introduced microRNAs, I said that microRNAs are like a brake on the system. MicroRNAs stop messenger RNAs from being translated into proteins. So then the question is, how would more LET7s which are a brake on the system, cause mitochondria to work better? Shouldn't they cause it to work worse as they get higher in levels? What was really fascinating is that paper that I mentioned that observed Argonaute binding to the 12S ribosomal RNA gene. They report that maybe microRNAs actually do the opposite thing in the mitochondria than they do in the cytoplasm. So I made this cartoon to adapt a figure in their paper, but what they report is that yes, in the cytoplasm, what we have always known about microRNAs is true. They prevent translation of proteins. They block ribosomes from synthesizing the protein from the messenger RNA. One of the ways that we know that they do this is there's an important protein cofactor called GW182. When GW182 is present, microRNAs block ribosomes from translating messenger RNAs. However, in the mitochondria, we don't see GW182. It doesn't seem to get inside of the mitochondria. And so now you have an Argonaute without GW182, and this paper reports that maybe its absence now allows microRNAs to act as an enhancer rather than an inhibitor of protein synthesis. And here's just some data from their paper that show that this is probably true. Like I said, miR1 was the microRNA that they were studying. Here you can see that as they add miR1 to the system, they transfect or add it to the cells. The levels of this protein, which are two nuclear encoded proteins, go down. So from your mock and control to the two treatment trials, you have less of this protein. That's how microRNAs normally work, right? More microRNA, less of the protein target. But now if they look at mitochondrially encoded proteins, they see the opposite. When they add more miR1, the levels of this mitochondrial encoded protein go up. And this other mitochondrially encoded protein also go up when you add in miR1. And as a control, these three proteins, ND1, COX1, and COX2, are all involved in oxidative phosphorylation in the electron transport chain. So they said, okay, maybe it's just proteins that are involved in this process, but that is not true. COX2 is involved in oxidative phosphorylation, but it's actually encoded by the nucleus and then makes its way to the mitochondria. And you can see it behaves just like those normal cytoplasmic microRNA targets, where its levels go down when you add in this microRNA. So all of the evidence they put forward in this paper, just over 10 years ago now, is that perhaps microRNAs in the mitochondria do the opposite to what they do in the cytoplasm because they lack this cofactor GW182. And if this is true, that would at least support the theory that I put forward that maybe LET7s, which are normally a brake on targets in the cytoplasm, could have the opposite effect inside of mitochondria, where when LET7 enters and binds Argonaute to a target, it somehow turns on or accelerates protein synthesis. You remember that I mentioned that the most prominent target of the LET7 family was a ribosomal RNA, so we still need to reconcile how that's possible. How could Argonaute bind to a ribosomal RNA and then enhance the translation of proteins in the ribosome? That's a question that I'm still very interested in figuring out, but at least it's supportive of the theory that LET7 could enhance protein synthesis in the mitochondria and enhance mitochondrial function, and might fill in with those paths I showed or how it could be related to development and cancer. And so I will leave the story there for now. I'll just take a moment to thank a lot of people. So when I was in grad school, I was in Molly Meffert's lab in the Department of Biological Chemistry at Hopkins. Very grateful to have been there with some wonderful people. My fellow grad students, Emily, Jinbei, and Benita, were wonderful to work with. We had tons of undergraduates who came through the lab and visiting graduate students that I got to work with. And then currently at Mount St. Mary's, I'm just getting my lab started in my second year there. Madison and Nancy have been working on these projects. Madison has been working on seeing if we can improve SCRAP, that pipeline tool, to maybe process even more different data sets so we can pull in lots of information to find new patterns. And then Nancy is looking to see if we see these signatures of LET7 binding to this ribosomal RNA in other species. All the data I showed you was from mouse, so she's been mining to see are any human data sets out there and other model systems that we can study and look at. If you're interested, this QR code takes you to our lab website where you can learn about the stuff we're doing in the lab and the students who are lovely. But with that, I'd be happy to take any questions. I appreciate you guys listening to my talk.
That's awesome. Thank you so much. Anybody feel free to unmute yourself, or you can also feel free to put it in the chat and I'm happy to read it for you. I'll start off. I've got a couple questions. I'll ask the obvious question. When you express LET7, what happens to the RNR1 level of RNA? I mean, you'd expect, right? If
Yeah,
this
that's the experiment we want to
microRNA
do.
binds to
Yeah.
the RNA should go down.
Yep, that's the experiment we want to do. So there's a couple of things that make that experiment difficult. For one is it's hard to sequence ribosomal RNAs. They're so abundant. Most companies, when you send your sequencing data, the first thing they do is remove ribosomal RNAs from the data because they contaminate. They're so abundant. So that would make it difficult to do by sequencing. And we've like asked companies like, is there a way you can do this? And it seems hard. You could do probes and run them on a gel and see. Not something we've looked at yet, but that would be the easiest thing to try. So unfortunately, I don't have an answer. We have not yet looked at just transfecting in LET7 and seeing how levels of RNR1 respond, but it's the experiment we want to do next.
Well, let me follow up and then I'll let somebody else get in. How do you think, I mean, so this is a very abundant
Mhm.
mRNA, right? And so do you think that this microRNA is really going to affect the expression in, I mean, do you see this as being like a tweaking of the levels of something that's already so abundant? Or do you think it's actually having like a significant
Yeah.
impact on the abundance and overall translation?
It's a good question. One theory I've had is that maybe what it's doing is not actually impacting the levels as much as what the ribosomal RNA is doing. So for a normal messenger RNA, right, it does two things. It blocks translations and can also lead to degradation. So it is minimizing their levels. What I have thought of is maybe that when Argonaut binds to the ribosomal RNA, maybe it facilitates or helps assemble the ribosome, right? When the ribosomal RNA gets synthesized, maybe the part of assembling that enormous ribosome is a LET7 facilitating that some way. And I've tried to look at crystal structures to see where the binding site is in the ribosome. Is it exposed on the outside or tucked away on the inside? The closest I could get to looking at this is it seems like it might be exposed. It seems like this binding site is not tucked away in the complex. It seems to be maybe on the surface. And I'm only about 50% sure of that. I had to look deeply. But if that's true, that would maybe give me reason to believe that it's somehow involved with ribosome assembly or ribosome function, not at the level of regulating the levels of the ribosomal RNA, but at ribosome assembly and function.
Cool. So in the chat, what triggers the increase in LET7?
It's a good question. So there are transcription factors that will collectively enhance the expression of these. One of the processes that my grad school lab studied is how Lin28 regulates their levels. So one of the mechanisms is that in the nervous system, I can give you that example that we studied, LET7 gets transcribed, so you make the precursor LET7, and in order for it to process, it has to get chewed in that last step to become the mature microRNA. The protein Lin28 interacts at that step, and so it can bind and prevent the LET7 from being recognized. So one way that cells increase levels of LET7 is by degrading Lin28. And so my grad school lab showed that when a neuron gets signaled, there's a signaling cascade that leads to the phosphorylation of Lin28, and then it gets degraded. Now there's nothing stopping LET7 from developing. So that's one mechanism that the cell uses, is by getting rid of Lin28 and freeing LET7 to be induced.
Thank you.
Yeah, no problem.
The mitochondrial ribosome significantly different than the normal eukaryotic ribosome.
It's a good question. They are different. They're different sizes, different sequences, different proteins, so they are quite different. Is it possible that it predates the formation of eukaryotic cells? It's a very interesting question. As far as I can go to say is that the mechanism of post-transcriptional gene regulation by microRNAs is a eukaryotic phenomenon. So we don't see this system or really anything like it in prokaryotes. There might be something tangentially related, but this seems to be a eukaryote-only phenomenon. So I would say probably not for a prokaryotic precursor. But what I think is fascinating that I don't have an answer for is I'm teaching my students biology now, like the endosymbiont theory, right? That maybe mitochondria were engulfed by a primordial eukaryotic cell. If that's true, how did the LET7 sequence that's theoretically in these genomes start to adopt to the mitochondria? Is it just by chance that there was a compatible sequence in there? that to me is a really fascinating question. So for your parts, they are quite different for the ribosomes in the cytoplasm and the mitochondria, but I don't think we see this pre eukaryotes.
Other questions? So I'm hearing a lot now about kind of spatial single cell sequencing, but also spatial sequencing. Do you see that kind of technology development as being applicable or helpful here?
I think it is helpful maybe to answer questions related to this. From my understanding of those techniques, the sequencing is good for messenger RNAs, like seeing where things are located messenger RNA wise. I'm less sure that spatial sequencing would work on these smaller RNAs, but I think what would be really cool is to see where these messenger RNAs in the cell related to what LET7 is doing to them. And so one theory that was explored is the idea of local translation, like in neurons, that for a neuron to respond quickly to an external stimuli, maybe one of the fastest ways to do that is just to ship all of the messenger RNAs out to the ends of the neurons, have them sitting and waiting, and then when the signal comes, you then release those RNAs and do translation, and now you have your proteins where you need them. So I think where spatial sequencing or spatial evaluation comes in is maybe seeing where those RNAs, messenger RNAs are, and if it were possible to see when they get released, like when do they start being translated in response to signal. And one of the mechanisms people have proposed is what holds them in waiting could be a microRNA. So a microRNA holds the messenger RNA out in the distal process, and then when the signal comes, the RNA is released, messenger RNA is free and gets translated. If that sequencing, that spatial technique was able to resolve, like bound, unbound, free can now be translated, I think that'd be a really cool application of what microRNAs could be doing in distal compartments. Yeah, that's a cool idea.
Any last questions? Don't be inhibited. Any level of question is good.
Yes.
Okay. Well, then this was awesome. Really exciting, interesting stuff. Thank you
Thank
so
you.
much.
And if you guys want to get in contact, please, you can scan the QR code, send me an email, connect on LinkedIn. I'd be happy to chat more about it.
Awesome. A reminder, everybody, we have another seminar next week on fMRI imaging and schizophrenia. And then we have another Science Slam coming up in early March. So please come back, join us for those things. Yeah, the recording will be available. I'll post it probably tomorrow on our seminars page. We'll have it posted there. So please share it out. Tell people how good this was.
Thanks, everyone.
Thank you, everyone. Good night.